And we are now recording, and thank you very much for giving a Build a Cell seminar, and it's all yours.
Okay, share my screen. There it is. Bring in my PowerPoint, and here we go. Okay. So yeah, thank you everyone for being here and for people online for seeing this afterwards. Today, I'm going to show you some of the work I've been doing over the last few years as a postdoc at Stanford University, and a little bit of background on myself. Before the pandemic, you would have seen my head down in this window. That's where I would spend many hours every week, and I haven't been here in a while, but I miss it terribly. This is the Stanford's bioengineering department, and I am a part of the Wholesale model team in the Marcus Covert's lab, where they were trying to build some revolutionary new technology that allows us to model whole cells. This is the team that in 2012 published the first whole cell model of Mycoplasma genitalium. And since then, we've been working on modeling E. coli with the same way. Some of that you'll be seeing here. Okay, so to start the presentation, first I want to state the challenge that we are addressing with the technology I'm about to show you. So this is actually a cartoon I saw Zoltan share a couple of weeks ago, and I thought, oh, this is perfect. I hope that's okay if I reuse it, Zoltan. So this depicts a parable, an ancient Indian parable of the blind men trying to make sense of this elephant. And each one is feeling a different part of the elephant. One of them back here says, "This is a rope." Another one up here feels another part of the elephant, says, "It's a fan and a spear. And here's a snake and a tree and a wall," and so on. And so the analogy I want to make is, as computational biologists, these blind men would be actually pretty spot on with what computational biology does. Computational biology often looks at different parts of a system of interest and builds a model, where it might be a stochastic model, a Gillespie algorithm over here, on the top left. A lot of people use flux balance analysis. It's a constraint-based approach to modeling, or you might go into the full like molecular dynamics, or multi-cell systems. And well, the truth of the matter is that none of these is capturing the biological system in how it really is. We want a model of the elephant, and if we capture different parts in isolation, we're missing something very fundamental about biology, which is that it's multimodal, with mechanisms operating across different scales, and they all come together to make the biological system what it is. And so this is the challenge we face. We want to bring all of these things together to make sense of our system. Now, what the software, I'm going to show you a little bit of software. So this might be a bit different from a typical build a cell seminar. I will be reducing the computational talk as much as I can, and make use of diagrams such as this one and minimal examples. And so Vivarium is the name of the software. It's a flexible model interface. By analogy, we've been inspired by what TCP/IP provides for the internet. It's a protocol that lets anyone make a system or a web page in the case of the internet, a model in the case of an integrative model. And so anyone can do it and plug it in and into this large, complex, and open-ended network. So our goal is to make it as easy as possible to define a dynamical model, here depicted with in orange, these orange rectangles. And you can define it, whatever it is that you might think the relevant mechanism is, and plug it into an existing system or build a new system and simulate the whole thing in a multi-scale representation. Okay, so I'll show you a little bit about how this is working for us and the progress we've made. An outline of this talk, we're going to start off with making a process of minimal transcription. Now this is simplified for the sake of demonstration. So so I hope the biologists and chemists might take it easy on me for this, but the goal is to make it as realistic as possible. And in the later examples, I'll show you more realistic models. Okay, so we start off with a minimal process, then we make a composite, which, as I'll show you, is how things are combined together. We're going to add complexity. Part of the goal here is to allow for iterative design, that you can add and layer on complexity in your model. We want that to be easy, so we're going to show some of that. Hierarchical embedding, you can place models within models within models, and I'll show that with division in an environment. And finally, some more examples of what we've been doing with this. Okay, so the basic elements of Vivarium are processes and stores. These can be thought of as kind of computational support for the basic difference equation that you can see over here, in which you have an update function here, an f, that takes in the state variables and the parameters with constants, and returns an update to the state. So a process, and this is the main star of the show, it's what in computer science is called an actor, that it can run on its own thread of execution. This one takes in the parameters. We also have these things called ports, which allow it to connect up. And the update function is the model, the dynamical model of whatever mechanism it is you might be interested in. Stores are databases that hold the state of the system. So they hold the state variables. They also hold additional methods about how to handle those states, how it's updated, how it's divided, and different properties. Okay, and a basic simulation loop, which it gets more advanced than this, but you'd have the process update the state in the store, and then it gets a view of the new state, and you go around and around and around like this. Now you can also make it parallel, so each process can run on its own thread of execution, and then this communication is handled by message passing. So we've been able to run these simulations on the cloud with hundreds of CPUs, and it can get truly massive in scale. Okay, a very minimal example that I promised you, and this is about as extensive as we're going to get with the code. What we call a process interface protocol has 3 parts to it that need to be instantiated for a process. You need a constructor. Here you see this is the constructor, the init. You declare default parameters, and these can be then modified when you initialize by passing in new parameters that will override the defaults. So here with this transcription process that we're calling TSC, you have a transcription rate and a degradation rate, for example, but we could pass in new parameters to override that later on, and it's really easy to modify. Okay, the next part of the protocol is a port schema. this declares how you can connect to this process. So we're going to have an mRNA port and a DNA port. In this minimal example, the mRNA, we're just going to have a single mRNA in there called C. And DNA, there's going to be a single DNA or gene called G. And you also, here's how you declare the properties of this state. So like a default value, how it's updated, how it's divided, various properties. This thing is very, very modular, so you can load in entire functions in here to declare how this thing's going to unfold. And finally, the last part of the interface protocol is an update method. You start, the update method takes states and a time step. You retrieve the state at the start, you run, here's our mechanism. This is just a simple difference equation. And then you return the update through the port. So we retrieve G and C through our DNA and mRNA port. We get our change to C, the RNA, and then we return our update to C through our mRNA port. We can plot out how these things are connected. So the transcription is connected to DNA. This is almost overkill because it's so small, but we have a different store for DNA and a different store for mRNA. And then when we run it, you see that our mRNA increases for some time until it hits a steady state. GNA is not being updated here, so it's just going to stay constant. Okay, next part, we're going to connect some things up. To do this, we declare what we call a topology, which is a bipartite graph of processing stores. So you can hook up multiple processes together, and where they overlap within the stores, if they have shared variables, that's how these processes communicate with one another and influence each other's dynamics. So let's see an example of how you make a composite. Okay, so here we have that previous transcription process that I showed you, and we're going to add a translation process, and we're going to connect it up to the RNA. So the constructor for a composite, now this is what we call a composition protocol. It has 3 parts as well. The constructor, you can pass in and override any of the processes parameters at this level. We have a generate processes that you initialize your transcription, your translation process with new parameters, and you return it. And then this is how you connect them up. So really, really simple way to connect things together. Our transcription process, its DNA port connects to a store called DNA. You can see it succeeded in doing that here with this output. mRNA is going to connect to a store called mRNA. And then our translation process, mRNA is going to connect to the same store called mRNA. It's in the same namespace. And our protein port for translation is going to connect to a protein store. So you can see what this all means here. We run it for some time, our mRNA increases, and then protein is expressed. And there you go. You have a composite model of transcription and translation. Very, very simple composite model. Okay, we're going to start adding a little bit more complexity and use this opportunity to share the interesting and exciting features of Vivarium. So Vivarium supports multiple time scales. Each process can declare its own time scale. And as you can imagine, processes in biology vary widely in their time scales. And so the process can change these things dynamically, or you can just declare it upon initialization that this thing operates in seconds or milliseconds or microseconds. And this is kind of how this is a picture of how the algorithm works. So at the start of a time step, a process gets the current view of the state. As it starts, it runs for its time step and then passes the update and reads the new state and then updates, etc. And here you have a slower process at the bottom that has a longer time step. And so it's going to read and write less frequently, but it gets the state at the start of the time step and then moves on in such a fashion as the temporal front proceeds in time. Okay, so we're going to take our previous example, and now we're going to add a little bit of complexity. The transcription process is getting swapped out for a stochastic transcription process in this case. I'm not showing all the details here, but this I can tell you this is a Gillespie algorithm here that's running, so it connects to our DNA, and in this case we have counts because Gillespie works in counts. So there's a way to add these auxiliary helper processes that do conversions. In this case, we have a process called concentrations that takes our counts and converts it to concentrations, and then that's what the translation process plugs into. Here's how the variable time steps are calculated. So there's this function. This is you can override it in your process to determine the next time step. Here, just a basic Gillespie algorithm determines how long until the next time step, until the next update. And when it runs, so here you can see this is in the concentrations that are being put out, and can't really see that there are variable time steps, but you'll have to take my word for it. So things are being updated with variable time steps, whereas translation is still being updated with the original time step. So here mRNA is kind of doing its stochastic thing, and protein is being expressed from that mRNA. So we have a little bit more stochasticity. Okay, stepping it up a little bit, let's add more complexity. I think there might be a chat. Benny, is the model volume dependent, as in, can you control for cell size? Yeah, absolutely. We can control for cell size. Now, Vivarium itself is not a model. It's kind of a scaffolding for a model. So if you declare a volume variable and your process reads that volume variable and uses it in the calculation, then you can use volume. And in this case, we do have volume. So we've part of what you're seeing here is really separating out the interface of the framework from the framework itself. So the model is as good as you make it, there's many reasons for doing that, but we don't provide the specific model, although there are many that you could use. I'll tell you a little bit about that later. Okay, so adding a little bit of complexity, we can put models, we can do this embedding of nodes within nodes. So we use a bigraph formalism. This is a graph that has nodes that can be embedded within each other, which is in contrast with the more standard flat network that nearly all neural network models have, in which all the nodes are at a single level. And this allows us to support scale, and also, it's almost a biological truth that biology has compartments within compartments within compartments, and that embedding matters. Turns out it actually makes modeling easier if you organize things in this way. And so what you see here is you have a few processes connected to stores, and you can also have processes reach across the boundary of a compartment into what we call a boundary store, and that makes these things pluggable into each other. So here's this is called a place graph, or a hierarchy, in which the topmost node or compartment is, let's say, the universe or the environment in this system, and then you can arbitrarily embed nodes within it. So you might have like a cell, an organelle. You can declare how you want this to work. And what's nice about it is you can take a whole compartment and that becomes a pluggable element into another system. So you can model a cell and then try to plug it into different environments. Okay, also to support hierarchies, we need a different kind of update, which is hierarchical update. Now, what I'm depicting here, we have this simulation actually running. You can take different processes that do this operation for you. Hierarchy updates mean that processes, stores, and entire compartments can be added and removed during runtime. Again, I think this is something essential in biology, that that you don't have a fixed topology for the duration of your phenomenon of interest, but rather things are constantly being modified. And here you see the environment is on top, maybe agent one within it. Agent one has a process that triggers division, it says divide. And so here you see in the next time step, the environment has two agents, agent two and agent three. All the variables and processes inside of here have been duplicated and the simulation keeps on running. Next, A2 sends an update. There's a process that's called engulf that says I will engulf A3 now. And in the next time step, A3 is within A2. And so it connects to A2 as though this was, it can, if it were to be uptaking molecules, it would be taking molecules from within A2. And A2 would be taken from within A1. Finally, A3 bursts and all of its contents merge with A2's contents. And you end up once again with an environment and a single agent inside of it. And it might actually be the where you started in this case. Okay, so let's show you what happens here. This is the same model I showed you before, but now we've added division to it. And we've also added a process in the environment that calculates the mass of everything inside of it. So here's a little kind of a picture depiction. There's more processes here that I showed you before, but we're not, I drew this rather than generate it algorithmically. And so you have one compartment of that stochastic transcription translation model, and division makes triggers it. So now you have more, and they're all running in parallel. Over here, you can see what happens. So you start off with a single cell, it grows, and it divides, and then you have two. They grow and they divide, and you have four. And the stochasticity makes it that these things don't line up perfectly, and that's expected when it's stochastic. By the end of this simulation, you have many, many, many cells running in parallel. Now, this process, this colony process at the top, can calculate the mass of everything within it. And so you can see how the total colony mass is increasing in this exponential manner with a slight stochasticity coming from that process I showed you earlier. Okay, so multiple generations with growth and division. Let's see, I think there might be another message. Oh, Benny said thank you. You're welcome, Benny. Okay, let's see. How are we doing on time? We're doing all right. Okay, so that concludes the minimal examples that.
Hey, Eran, you actually have another question.
Oh, I do have another question. Okay. Oh, I do. Hi, Zainab. Is it possible to have two different cells after division? Are they identical by default? Yes, absolutely. It is possible to have two different cells after division. They are not identical by default. The states within it. So for example, here in our RNA case, this state got divided in half. So I guess these ones do start off with a similar state, and the protein gets cut in half as well. And so so the two daughter cells here are going to inherit similar states, but we can tell it to divide in different ways. So this, you might divide your state C just by divide by 2. Each daughter cell gets half, and in that case it would be very similar. But you could also choose different ways to divide. So you can have we have an example in which there's a front and a back. So we might have additional stores. If you see over here, this is a kind of a minimal case, but you could have a store that's like the front cytosol and the rear cytosol. And the front cytosol is going to go into one cell, and the rear cytosol is going to go to another cell. So it doesn't divide evenly, but it's already, you can include some spatial considerations. You could also, we have a binomial divider. Let me go back to show you what I mean by divider. So when you declare your process over here, this divider, you can tell it how you want to divide. This one is using a set. So in this case, the division is going to be equal. It's the whatever value the mother cell has, the daughter cell has. If I were to write split here, this is division, in which we're doing with the stochastic case. You could put in binomial, but you could also pass in a new function, so you don't even have to use the ones that are pre-configured that you can load in. You can pass in a function over here that does whatever it is you want upon division. It just needs to tell it how this variable is going to go into daughter cells. So the topology of the daughter cells at present with our way of division, the topology is going to be the same. But again, you can make it add new processes. So it's very modular, very configurable. You're welcome. Okay, and there's another question. Agnes, can you program evolution by defining some parameter like stochastic division of genome into daughter cells? You can. We haven't, but I've thought about it, and that's of course the ultimate goal of all of this. And so defining some parameter like stochastic division of genome into daughter cells, yes, exactly. So what you're seeing here, there isn't going to be any evolution with this minimal model, because the daughter cells both inherit exactly the same DNA and the same topology. But you could imagine, kind of going off of that, my previous response, you could have a kind of a stochastic or maybe a mutate divider that takes the state of DNA, which might be a sequence of base pairs, and actually mutates it, passes that on to the daughters. We haven't done mutate, but we've thought about it. I hope to get to that within in the near future. Okay, thank you. Okay, so now I'm going to show you some more examples, and I think we'll have plenty of time for questions after this. Okay, let's so some more examples. But first, something really important about what we're trying to facilitate here. We want to be able to start off kind of small with a few key assumptions and add complexity and layer on complexity and plug more processes in and switch it up and make it so that anyone can pull down a model from the internet. We've made it as easy as currently possible, but not as easy as ultimately possible to pull down models, define your own process, plug that in, see what happens. I mean, that's how hypothesis generation should work in our kind of computational theoretical domain that computational biology works in. You need to propose a mechanism. I think that transcription works, and this way, or that such a process is active, and try it out and see what it does. So here, this is a model that we published. I do have a reference for you in later slides, but we can kind of iterate to increase the complexity of the model. Okay, so first, to start off, this is an environment model that we've built. This is available for anyone to plug into. You can pull it down. It's a pip installable. You can pull down this environment. It has a multi-body physics process, which uses a physics engine, and it also has a diffusion process that diffuses fields of chemicals. It connects to two stores. A is the agents, which have locations, volume, length, width. Here I list all of the variables in here. And you have a fields store that contains the concentrations of a number of fields. Now you're not, I'm not showing fields here, but you'll see them later. Okay, and then you can have subcompartments here, A1, A2, A3. These are in this case, I've just plugged in minimal growth division agents, but they can be any kind of agents as long as they know how to connect to these agents and field stores. And again, going off of this iterate to produce complexity, when you build a model like this, you start off with your minimal assumptions about the agents, make sure everything's behaving as you like, and then once you're happy, you can add complexity. So here we're just kind of focusing on this. You can see I initialize the system with 3 cells that grow and divide, and over time we have many agents start to build up down here in the bottom, all pushing each other away. The physics engine is working beautifully. You don't see diffusion because these aren't growth division uptake and secretion agents. They don't actually touch the field, but you'll see that later. And so here you have a few hundred cells at the end, all running in parallel, growing and dividing, actually stochastically growing and dividing. And the physics looks good. Okay, now we're gonna add a simple, this is actually not even simple anymore, but compared to what comes next, it's simple. So this is a cell model. It has 3 processes. Metabolism uses a type of modeling framework called flux balance analysis. This is a paradigm for large-scale metabolic models, very popular. And you can see here the metabolism running, the external concentrations. You have some, so notice logarithmic scale is probably not the best way to plot it, but so you have some secreted nutrient waste products that go up, and everything else is kind of being depleted from the environment. And there's many metabolites that are just slowly increasing here. Again, logarithmic scale, so they're actually increasing quite a lot. And then our mass is taking the counts of all these internal metabolites that are building up, and we calculate the total mass, and it grows exponentially, doubling about every half hour or so. The next process is, well, we have a transport process. This is Michaelis-Menten style kinetics, and it connects to metabolism. You can see here V. This V store is fluxes. So it constrains metabolism through this store by defining the, in this case, lactose and glucose uptake rates that constrain the metabolism. And gene expression is an ODE with a bursting transcription leak. So that you can see here, first glucose is being taken up until it's depleted. At some point, due to this stochastic leak of transcription, you get some LAC-Y RNA being expressed, transcribed, and then LAC-Y protein slowly grows. And this is, the LAC-Y is a transporter that is going to bring lactose into the cell. So you can see that lactose starts to be depleted from the environment. And here with total mass of the cell, it starts off growing. Once glucose runs out, there's a lag phase in which it's not growing. And then lactose starts to be brought in, so it's growing again. And this is this is the result of everything being integrated in a model, in the composite model. Oh, and here's the reference if you want to read about this work. Okay, next up, we take that model that I just showed you, so this one, and we are going to place it in this one, and this is what happens. So we start off with 3 cells. Glucose is being depleted, and you can see that it's being depleted nicely. Here you see the field, how that all works. It hits a lag phase pretty early on. There isn't much glucose in this environment, so it doesn't feel much growth. So you have a lag phase in which they're not growing very much. Here you can see that the LAC-Y protein has begun to be expressed, and you already have 2 cells expressing LAC-Y. So probably somewhere preceding this particular snapshot, there was these cells' mother cell expressed LAC-Y, and they inherited it, and so they are able to start to take in the lactose field. You can't see it yet in this snapshot, but in the next snapshot that's being shown, the daughters, the descendants of these 2 cells, are taking over the population because they're able to grow. So you can see that LAC-Y is being highly expressed here. There's a little bit of a feedback cycle that once you're taking in lactose due to LAC-Y, the expression of LAC-Y is disinhibited. And you can actually see that the lactose field is starting to be depleted around here in the final snapshot for the lactose field. Okay, going to the full E. coli chemotaxis model. At this point, we have quite a few processes of truly multi-paradigm model. We have our metabolism with flux balance analysis, transport, Michaelis-Menten. This uses a template-based Gillespie algorithm for transcription and for translation. So we load in the gene sequence, and it's actually pulling out base pairs out of the pools, base pairs that have been created by metabolism. Translation, so complexation takes the proteins from the translation and makes complexes, which include flagella, and that uses the correct stoichiometry. So masses, in this case, mass is being preserved and being tracked throughout the whole system without any mass appearing out of nowhere. Okay, so degradation is ODEs. The proton motive force is the Goldman equation, which fuels flagella activity. And we have chemoreceptors, our Monod-Weyman-Changot statistical mechanics model of receptor ligand binding. And flagella here, you can actually see our subcompartments that so that they have their own state and their own mechanisms internally that create propulsion in an environment. Okay, a little bit of a taste of what is happening here. So here we see the chemoreceptors on their own. This is the Monod-Weyman-Changot model. That you can give it an external pulse. This is the ligand. In this case, it's a ligand called MeAsp. And you get a pulse of it. The cluster activity responds to that by dropping in activity or increasing in activity. Dropping in activity when the ligand is increased. And there's also adaptation. So methylation of the chemoreceptors makes it adapt to its external concentrations, so that responds kind of to relative concentrations rather than absolute concentrations. Okay, this is the flagella, which uses a veto model with stochastic switching. So you can see the external concentration of KY. This is part of the signal cascade downstream of the chemoreceptors and phosphorylated KY, and they're going through their stochastic dynamics. Flagella are here being added to the simulation during runtime. This happens due to expression complexation that flagella are being expressed. And the flagella have their own state, so they can either be rotating counterclockwise or clockwise. And the total state of the cell's motile state is a function of the sum of the individual flagella to be either in a run state or a tumble state. So runs, the cell is moving forward, and tumbles, it turns around randomly, and it generates thrust based on the activity of the flagella. So you can see here we place some, I think 12 cells on this green dot. Half of them have a receptor and a motor. By motor, just the flagella. And the other half don't have receptors. So there's no signal cascade that lets them respond to their environment. And we're putting a ligand gradient. I show you a linear gradient. This is actually exponential gradient with a lot of Mi-asp here at the right side of the environment. And so the cells that don't have receptors go through this random walk and don't get really far, constantly tumbling around. Whereas the cells that do have receptors are able to pick up on the gradient, and they have what's called a bias random walk. So they're moving up the gradient, still kind of twisting and turning, but overall make it towards higher concentrations in their environment. Okay, here's another project that we've been working on with Vivarium. This uses what we call a wholesale model. It's actually a half cell model. It's not complete. It's a paper we published in Science earlier last year. And Chris Skalnick, I've been working with this exceptional undergraduate student who has taken the wholesale model and plugged it into the lattice environment that I showed you up on top. So we're kind of taking this thing as wholesale in this case. And so you initialize it with a single wholesale model. Massive model, by the way. Each one of these is running in parallel on Google Cloud, and some 120 cores. Grows and divides. There's some stochastic expression. So you can see that by the end of the simulation, you have different expression profiles. This might go to Zeynep's question that the daughter cells aren't identical to the mother cell, and you can actually see that there's heterogeneous gene expression in this case. AMP-C, this is involved in the antibiotic response. You have some cells that are expressing quite a bit of it, and some that have none of it, are not expressing it. And this Acra-B-Tol-C is an antibiotic exporter, and likewise heterogeneous gene expression here. Okay, so this is going to be our second to last slide. And here, again, thank you to Chris Skalnick. He took the wholesale model, and using Vivarium, this is the same representation I showed you earlier of stores and processes, but he drew it a little bit differently than me. But he's adding all kinds of mechanisms related to antibiotic response, such that here's the wholesale model growing. Antibiotics are added here, and some of them die following it, and you can see the black cells are dying, And you can actually hear he drew the phylogenetic tree in which green cells survive. So antibiotics are injected somewhere along here after the gray dots. And some cells die, some cells respond, all due to how the mechanisms of response to antibiotics are active, and these heterogeneous expression profiles that I showed you in the previous slide. So those that are able to export antibiotics out of their system efficiently can make it to the next generation. Okay. So wrapping it up a bit, we all of this software is open source, and we've started a GitHub organization called the Vivarium Collective in order to build large models in computational biology. It's not going to be that it's not going to work if everyone works it on their own and makes a little model, but rather this to get accurate models requires collective effort, and we need to build off of each other's work and iterate and increase the scope of our models, testing hypotheses by proposing new processes, seeing how they work, trying out alternate hypotheses. You might have a different hypothesis about how things work and build a model, add it to the existing models. Does it make better predictions? And so the Vivarium Collective aims to support that collective effort. So please join the Vivarium Collective by add your process, combine it with existing models, and simulate it. You can we have been aiming to make more working groups on model integration, which it's been fun to try to integrate these things together and to think through it. There's also plans to make a workshop, which we've been calling Add a Process Workshop, or might be kind of like a hackathon, that we all get together with our mechanisms of interest and wire them together and try to run them. So if you're interested in organizing or sponsoring or just being part of something like that, please do let me know. You can check out the open source engine here in our GitHub, Vivarium Collective. We have a documentation and tutorial you can see over here, and of course, feel free to email me with any questions, and if you want to meet and propose a system that we can try to plug in. Please do email me. I live for this stuff. Okay, and so finally, thank you to the Vivarium team. These guys have been just outstanding to work with, and gals, and we have several collaborators that have used the Vivarium software for their own projects, and that's been very valuable to us to make sure that it works for everyone. The goal here is really any model should be able to go into this, and of course to the Build a Cell community. It's been a pleasure to be part of this community. So thank you very much. I see I have some questions. I'll go to those now. I'm gonna actually, let me go back and leave this up. Okay. Omar, okay, well, we're starting with Benny. Can you make models of multicellular organisms? In theory, yes. You could make a, let's go back to our hierarchy slide. Let's see if I can, how can I get to this? So Benny, in theory, yes, we haven't done it yet. I've of course dreamed of it, but you could, if you can make a hierarchy in which you have subcell types and they all interact with each other, and you know, with their own expression profile, then in theory, I don't see why not. This has been made to support really large simulations and different mechanisms. So I would love to move it in that direction. And I have a few thank yous. You're welcome. Okay, Agnes. Could we have build-to-cell workshops on using Vivarium to model synthetic cells with a pure system? Yes, absolutely. So we've part of the build-to-cell community. There is this synthetic cell project, the in silico cell, and Vivarium was very much inspired by me being in those meetings and kind of talking with people about what would be needed there. Kind of coincidentally overlapped with the Wholesale team's needs at Stanford, and so I've thought a lot about how to model synthetic cells with a pure system, and absolutely yes. I think we can do it, and I think we should do it. I've met with William Poole a lot. I think maybe the next build-to-cell workshop would be a good time to get that started. And bacterial TXTL instead of pure. We've been thinking about pure specifically, but sure. Bacterial TXTL. I mean, I think there's a lot of. I'm not sure actually the distinction there. Emma. I would be very interested to have a brainstorm about how to do that specifically. Okay, moving on to Damien. Related to the question about the multicellular model, I guess you could also then make models of things like bacteria consortia, right? So this is like a microbiome-like question. I take it, Damien. If you have different bacteria all interacting in the same model, what happens? Yes, let's do it. One of the models that I've been that's been built in Vivarium looks at tumor and T cell interactions. So not exactly bacteria consortia, but you have two different cell types, tumors and T cells, and based on their own expression profiles and where they're placed spatially, and how many of each there are, we get different outcomes, whether the tumor grows and overwhelms the T cells, or whether the T cells are able to get it under control. And I expect kind of a similar behavior. Maybe with bacteria consortia, you could have a lot of cooperative interactions that could really help. And I did show you. So something kind of related is over here. So you have these are all supposed to be E. coli, but you have different expression profiles where some are expressing LAC-Y, and so they're able to respond to the environment better than those that are expressing glucose when glucose has been depleted. I would love to think about bacteria consortia more and see what we can do about that. And so Kate, yes, a workshop on chemical reaction modeling, that link over there. I'm not sure if this will be available online, but maybe we can add it in the description or something. This was an outstanding modeling tutorial back in the fall, in which it was six lectures. The first mostly focused on chemical reaction networks, and then in the final lecture we looked at Vivarium, and there's some more content there if anyone wants to find that on YouTube. That there was an actual coding section that we were able to go in and model things with Vivarium run them. So for people on YouTube wanting to find it, there's another good link on Build a Cell to a slightly more technical Vivarium talk. And that makes it to the end of the questions.
Thank you very much. I really appreciate not just the explanations of the tutorials, but the background you gave. I think that was really useful for a lot of people.
Yeah, glad you think that. That's why I made it. Most of my talks are have a lot more code and more aimed at computer science, but it's really important to provide examples that people can understand. So I'm glad you think so, Kate.
And thank you very much, everyone. And if you have any more questions, you can look up Eran's email, and see you all next week.
All right.
Thanks again. Bye.
Bye.